All right, thanks. Thanks, Frank. So my name is Barbara Weiser, and this is my second year in the RET program. And my curriculum is on intermolecular forces, and it's called There's a Thin Line Between Attraction and Repulsion, Understanding Intermolecular Forces with TLC. I work at Santa Paula High School, and I teach chemistry honors, chemistry CP, and I'm also a teacher in the Agriculture Academy, and I teach agricultural chemistry. I've been at this will be my 5th year going at Santa Paula High School, teaching chemistry, and like Frank said, I've taught for about 10 years. Santa Paula High School has about 1,500 students. It's about an hour away from UCSB. It's a really pretty campus, and I really enjoy teaching there. All right, so last summer I did my research experience in the Ecology, Evolutionary, and Marine Biology department at UCSB, and I was able to work in the Oakley lab, and my mentor, my amazing mentor who's here today, thank you Emily, is Emily Lau, and she is a chemist working in an evolutionary biology lab. And the big question that they're working on, that she's working on in her projects are, how did ostracods evolve the ability to produce the bioluminescent substrate luciferin? So it's an evolutionary biology lab, so they're really looking at all this, the different evolutionary pathways that this luciferin and bioluminescent molecules took. And the smaller research project that we're working on are what small molecules are unique to luminous ostracods. So this is a picture of an ostracod, and it is giving off this bioluminescent mucus. And this is the chemical equation of how bioluminescent light is produced in an organism. So you have this luciferase or another photoprotein, and then luciferin, which is a small molecule and reacts with oxygen to produce this bioluminescent light. So we just looked at a small aspect of this because it was just one short summer. And the procedures that we used were thin layer chromatography and cross-reactivity. So we added the protein luciferase to a small molecule luciferin, and we tested the presence of luciferin and if the organism was producing bioluminescent light using thin layer chromatography and cross-reactivity. So the results of the thin layer chromatography are over here, and they're showing the presence or absence of different molecules. And then the cross-reactivity showed that bioluminescent light was produced in bioluminescent organisms, and it was not produced in our one non-bioluminescent species of ostracod that we were testing. So this was a really fun lab, and it was really cool because bioluminescence is really cool, and it really is seen in a lot of different organisms that I wasn't even aware how many organisms that bioluminescence occurs in, and all the different evolutionary pathways that organisms use to have bioluminescence, and a lot of them are very different. So it's really cool. And I was also excited because I really enjoyed doing the thin layer chromatography, and I felt I haven't done that in a really long time, so I felt like I was learning it again, and cross-reactivity was a new technique that I'd never done before. And so that was, it was really neat to learn those new techniques or relearn a technique that I hadn't done in a long time. And we don't normally use thin layer chromatography in our labs, but I thought that was a really nice one because it's pretty simple, straightforward procedure that I think the students could do, and also the materials are not are accessible. They're not too hard to come by. So I was excited that to learn that again, and I feel like it's something that I can really apply to my classroom. And I also decided to use thin layer chromatography and chromatography in general, because I also use a lot of paper chromatography, but I think it will apply to a lot of the careers and interests that my students at Santa Paula High School have. I have a lot of agriculture students and students that want to go into the medical field, and students that want to go into all sorts of different industries that use chemistry. And it seemed like chromatography really can be used across a lot of different industries. And it's a good chemistry technique that I think students will be able to use in any, if they go into any kind of science or chemistry related career. So some of the connections between last summer and this summer are intermolecular forces and polarity and chromatography uses in chemistry. And again, my inspiration was just that students can use this in agriculture, medical fields, or other industries. And a lot of my students have a lot of different interests. And again, you can use it across a lot of different disciplines. Some of the challenges that I feel I will face are just teaching the techniques. While it's a relatively simple technique, I think just at first, it's going to take a lot of practice and a lot of time to teach it. And then, of course, the vocabulary and the language. So any science is going to have a lot of different vocabulary and words and concepts that students aren't familiar with. And again, just a reminder, my students are chemistry students. They're juniors and seniors. And so a lot of them are starting to think about what they want to do and go into industry or applying to colleges and things like that. So I think learning this technique and all the different applications are going to be helpful for a lot of them in the future. And I teach honors, CP, and ag chemistry. So my unit plan is based on the 5E model. And so we use this as a science department. It's our standard model that we use. And so I have developed the 5Es, and you can see probably already I added an E. And so these are the aspects of the unit that I have planned out for the intermolecular forces unit. So the Engage is going to have a candy chromatography lab, and then in that I'm going to do a few food coloring demonstration just to demonstrate how the students can do the techniques. And the Explore will be an ink chromatography forensics lab. And the Explain, we're going to use previous materials and modify those for the unit. And then the Elaborate will be a chlorophyll chromatography lab, an over-the-counter medicine chromatography lab, we'll incorporate the bioluminescence research from last summer with a video, and then a short lesson encompassing that. And then finally the Evaluate, which is the ink lab testing different solvents in chromatography. And then the Extend is something that I'll explain in a little bit that I hope to use in a future unit. So the objectives of this unit are that students will learn how polarity of molecules creates intermolecular forces, how intermolecular forces cause bulk properties, and bulk properties are things like density, temperature, boiling point, melting point, capillary, we're drawing a blank right now, so that just capillary action and vapor pressure and things like that. And so those are bulk properties. Bulk properties are things that you need a lot of molecules in order to see the property, and not just one molecule or atom. And they'll hopefully learn in thin layer chromatography, applications chromatography in industry. And then the standards that this unit will meet are using labs and lessons to compare the structure of substances at a bulk scale, and then just looking at how electrical forces between particles cause intermolecular forces and electrostatic forces to create polarity. So these are the 3 activities that I focused on over the summer. They're in blue: the candy chromatography lab, the ink chromatography forensics lab, and the chlorophyll chromatography lab. The other ones are still a work in progress, or I'm going to just use and modify things that we've already developed over the past few years. So the candy chromatography, as you can see, we're going to use M&Ms, and this will be an introduction to polarity and intermolecular forces and an introduction to chromatography techniques. So at this point, this will be the first lab where students use chromatography. So a lot of this is just going to be showing how to use this technique, and then just a basic introduction of intermolecular forces and polarity. So our question is, why are there 10 dyes used, but only 6 different colors in M&M candies? So here I've circled the dyes that are used in M&M candies, and you can see that a lot of them, there's really only 3 colors that are used. It's 10 dyes, 3 different colors: blue, yellow, and red. So how are we using 10 dyes, but only 6 colors there's only 6 colors. So the techniques that we're going to use are dye extraction, and we'll just extract the dye from the M&Ms by soaking them in water, and the colors will dissolve into the water. And then paper chromatography techniques and analysis. So that would involve spotting the plate and then running the plate with a solvent. And in this case, it's a 1% solution of NaCl. And then later analyzing the developed chromatography paper. And then so this one will just be comparing the different dyes, and where we're going to talk about polarity is the dyes that travel farthest are less polar than the dyes that travel the least. And then finally, the end of this lab, we're going to predict polarity of molecular structures. This is a kind of an advanced activity, but I thought it would be interesting for students to just sort of start to look at these molecules and see where they see negative and positive charges, or any kind of aspect that would create an electrostatic force and thereby an intermolecular force. And so that we'd be kind of predicting which one might be most polar. And then we can go back and use our results from the chromatography lab and compare which ones are more polar than others. So we can research some of this already published data and these molecules that have been established, and see if we can compare the polarity, make a prediction, and then compare the polarity later. So industry applications for this lab would be food science. And this should take about 1 to 2 days. So maybe one day to set up the plates and then another day to run them. So the Explorer, which is the next E in the 5E model, is the case of the missing cupcake. So this is a forensic chemistry lab. And this would be, each of these labs have learned in the previous lab. So in this one, they will practice chromatography techniques using ink chromatography, and then we're going to introduce the retention factor, and that's a more quantitative comparison of the different molecules that are separated, or different compounds that are separated from a mixture. So in this case, one of the teachers will be implicated, one of four to five teachers will be implicated in stealing a cupcake from my desk at school and holding my cupcake ransom for some cookies because they prefer cookies. So, so the students are going to help me determine which teacher took my cupcake. So because I don't want to have to go out and buy some cookies for them. So we're going to try and figure out which teacher stole my cupcake first. And so what we're going to do for that is so the questions are, how do we determine which marker was used for the ransom note? So the ransom note was written with a marker. I'm going to go to each of the teachers who I think may have taken my cupcake and get a pen or get a marker. And then we're going to try and figure out which marker was used which marker was used for the ransom note. So the questions are, how can we determine that? And then how can we analyze mixtures by separating them into compounds? So this is going to use a paper chromatography technique, and then we'll be identifying an unknown sample based on comparison of known samples. So I have 4 different markers, and then we'll use the chromatography to compare the type of marker. And then from that, compare the ink from the ransom note. So this one plate, the smaller one with just one, that's the ink from the ransom note, and then compare it to the 4 different markers that I have collected. And then in this one, we're going to introduce the retention factors. So we're going to use the RF values to accurately determine components in the sample. So, and from those 2 things, we're going to determine the cupcake thief. And so as industry applications for this would be forensic science, and this should take about a day. So the first elaborate that I had time to develop over the summer is a chlorophyll lab. So in this, the objectives are to learn, so this would be learning thin layer chromatography techniques. So this is going to advance what the students know up a little bit from paper chromatography to thin layer chromatography. And thin layer chromatography is more accurate, and you can get sharper contrasts, and it's just a more accurate kind of technique and a little bit faster than paper chromatography. So just a little bit more advanced technique. And then we'll apply the concepts of biochemical functions of plants and photosynthesis. So at Santa Paula High School, students do biology before they do chemistry. So they'll have already learned photosynthesis. And so this will kind of be looking at that in a little more detail of the different chlorophyll pigments that there are in plants. And the scientific inquiry question for this would be, how can we compare chlorophyll pigments present in different types of photoautotrophs? So photoautotrophs are organisms that produce energy from light, and not all photoautotrophs are plants. So blue-green algae would be one of the things that we test. So the learning techniques would be chlorophyll extraction techniques and thin layer chromatography techniques, analysis using RF factors, which they've already learned the RF factors, and then compare the presence of different chlorophyll molecules, and then also compare those to published data where we can determine which color corresponds to which pigment. And then so the first thing was I'll choose organisms to analyze for chlorophyll. So there's a few different methods which I might ways that I might follow. For now, it just tested spinach, blue-green algae, green bean leaves, and kale. So the first thing we did, the way to extract we could extract chlorophyll or use prepared powder. And there's a few different ways to extract chlorophyll. The method that I used over the summer was just to grind the plant up and then dissolve it in acetone. And then there's a few other methods too that can be used. And then some of the substances I did use a prepared powder. So the blue-green algae, I used a prepared powder. And so then we use thin layer chromatography to separate the chlorophyll pigments. And you can see that they're separated down here on those plates. And then compare the presence of different chlorophyll molecules. So here you could see that blue-green algae just has the one, possibly two pigments here, and then kale and spinach and the green bean leaves all have more pigments involved. And then so at the end of that, we will determine the chlorophylls, the different types of chlorophyll present using RF values from samples and then published data. So this industry application for this would be agricultural science, and that time for this would be about 1 to 2 days. And then the next elaborate, which I'm still developing this one, would be testing over-the-counter medicines. So this would be, again, practicing thin layer chromatography techniques, using chemistry techniques to determine compounds present in over-the-counter medicines. So our question for this activity is, how can we determine compounds that are present in certain over-the-counter medicines? So they're going to go back and use techniques they had already learned. And then in this case, we're going to add using a UV light to visualize colorless compounds. What's nice about the chlorophyll lab is that the compounds can be seen easily without just with the naked eye. But with other things, a lot of compounds are colorless. So we'd use a UV light to determine where the fronts of the molecules are on the thin layer chromatography film. And in this one, we'll research published data to determine compounds present in each medicine, and then finally use a known standard to determine unknown compound or medicine. So they'll test things like aspirin, maybe no dose with caffeine pills, ibuprofen, acetaminophen, things like that that they might see on a normal basis. And then I'll give them one unknown, and based on what they've seen with their plates and the retention factors, they should be able to determine what that unknown is. So I think that'll be pretty fun. And the industry applications for this would be pharmaceutical science, and this would also take 1 to 2 days. And then finally, the final piece of the elaborate would be the bioluminescence research video. And so again, my mentor is going to make a video based on her research and how TLC is used in that. And I'm really excited about this because this will show just another aspect that students can use TLC for and other chemistry techniques. So this one would be the industry application for this would be academic research. And the students would learn about applications of chromatography in current scientific research, and then also learn about evolutionary concepts and how chemistry is related to evolutionary concepts. Okay? And again, they learned a little bit about evolution in biology. And this would take maybe half a day to a day. And then finally, the evaluate is where this is the part where the students already should have an understanding of polarity, intermolecular forces, and bulk properties. And they're going to use that, and they're going to just, they're going to test, we're going to go back to the ink lab, and they're going to test different solvents to determine which solvent is the best or most effective in creating a good chromatography plate. And this should test the skills that they've already learned, right? With something that they haven't really done yet. We haven't really tested different solvents, but they've already seen this lab, so they can go back and test something new about it. So again, and this was also inspired by my research last summer, because when we were going through and trying to run the plates, Emily had tried a few different ratios of solvents to determine which one would work best to expose the chromatography film. So I thought that'd be something interesting that they could do. And also they could do a design project with this one and design and modify their own experiment that could test these different solvents. And they determine the most effective solvent for use a chromatography procedure. So industry applications for this would be any chemistry field, and this should take about a day because again, just spotting those plates with an ink pen is pretty fast. And then finally, Mike, the extend, which is my 6th E that I added. I think this would be really cool to use later on when we do chemical reactions. We'll revisit the procedure and students should be able to run a plate to see if a reaction is in progress and if a reaction has completed. And I think that'll be a good way to visualize new products that are being formed from reactants. So they would, again, design and modify this experiment using thin layer chromatography. And again, this could apply to any kind of chemistry field and should take about a day. So takeaways from this are the learning results would be that students learn concepts of polarity, intermolecular forces, and bulk properties in applied lessons, not just, straight worksheets and lectures and things like that, but also using these applications and techniques that can be applied to real life industry. And students will learn how to separate compounds from a mixture. They'll practice a chemistry technique enough times to become somewhat proficient, and then they'll learn industry applications of a fairly simple technique that's still used. And the total time of this unit would be about 12 days. With that, I'd like to thank the USB Materials Research Laboratory, RET program, Dr. Todd Oakley, who so graciously invited me into his lab to do this work, and my amazing mentor, Emily, who is I really learned a lot from her, even though we just, got to work together just a short time last summer. I really got a lot out of it. So thank you to Emily. And then to the program directors, Dr. Frank Kinnaman and Marilyn Garza, and then my cohort, Tori Poole. All right, with that, does anybody have any questions?